So, for those of you who are interested in how iron mismanagement can exacerbate the impacts of inflammation, how the over retention or the over absorption and over circulation of iron can lead to chronic inflammation, can lead to chronic disease, autoimmunity, I've got a little chemistry lesson for you. There will not be a test at the end. If you have questions, just leave them in the comments. We're going all the way back to Biology 101. So you have your cell membrane here with the nucleus where all the DNA is. You got things like endoplasmic reticulum and Golgi bodies making proteins. What we're focusing on here is the mitochondria, the mighty mitochondria that your science teacher was talking to you about. The mitochondria is where cellular respiration happens. This is how the body basically makes its own energy from the food we eat and the air we breathe. And it turns into the carbon dioxide we exhale with every breath and this beautiful molecule called ATP, or adenosine triphosphate. ATP is the energy currency of the cell. It's how all of the things that happen within every cell actually happen. And that's where people's understanding of this whole process usually comes to an end. But if you went just one level deeper, you can see where inflammation could potentially come from. Because if you looked at the electron transport chain, which was actually creating the electron gradient to instill this energetic output into the energy currency, you'd see a lot of really interesting ingredients. So I'm going to skip a lot of the details here, but the electron transport chain is a series of proteins and enzymes where hydrogen ions and electrons are being transferred from molecule to molecule. And at each step of the way, it is gaining energetic potential until eventually a phosphate group is added to ADP to make the final product. But with this transfer of electrons along the way, you're going to need specific molecules that are able to accept and donate electrons along the way. And it would be no surprise that iron is that molecule, that electron donor and acceptor that is facilitating the movement of this electron along the gradient and to the final product. Every single complex of the electron transport chain has a significant volume of iron for the reason of creating energy. And just as energy can't be created or destroyed, all of the chemical reactions that are happening through the electron transport chain with the handing off of electrons, they have byproducts. And those byproducts, not coincidentally, are reactive oxygen species. So what are reactive oxygen species? The three main ones in the body, in order of least reactive to most reactive, are hydrogen peroxide, superoxide, and hydroxyl radicals. So as these electrons are being transferred from molecule to molecule, creating more and more energy along the way, at each of those stages, when that electron gets transferred, if one is being transferred, there's naturally going to be something that it gets transferred from. And so with all of these electrons and the resulting electron-less molecules, a lot of those end up as reactive oxygen species. And it would be no coincidence that the mitochondria are one of the greatest sources of ROS within the cell. Now, it's important to note that in a typical electron transport chain reaction and creation of ATP, the ROS of choice is hydrogen peroxide. Why? Because it's the least reactive. It's the most inert and less likely to react with any of the other things going on in the cell. So let's recap real quick. The mitochondria is taking in glucose and creating cellular energy through the mitochondria, of which the byproducts of that reaction are ROS, facilitated by the natural electron acceptor donor activity of iron. So this relationship brings me next to both Fenton's reaction and the Haber-Weiss reaction. Fenton's reaction is where an iron molecule, an unbound iron molecule, reacts with hydrogen peroxide and through a series of steps becomes a hydroxyl radical and a superoxide ion. And this is super important to note. You are taking the least reactive hydrogen peroxide ROS, reacting it with an electron donor or acceptor, and as a result, you are creating 2 out of 1 more reactive oxygen species. The Haber-Weiss reaction is a secondary reaction to some of the byproducts of the Fenton reaction, in which another unbound iron molecule reacts with a superoxide molecule and through a series of steps ends up with an additional hydroxyl radical. So what was first just one relatively inert ROS has now, through 2 separate reactions with free iron molecules, turned into 2 of the most reactive reactive oxygen species. This whole situation here is absolutely terrible news for the cell. And you may be wondering to yourself, well, if this is so terrible, why the hell are these 2 things here? And that's because the mitochondria has a natural defense system to quickly neutralize these hydrogen peroxide byproducts into manageable, disposable things. Those natural defenses are these enzymes: superoxide dismutase, catalase, and glutathione peroxidase. These put the kibosh on these. So in essence, the creation of every single ATP out of mitochondria has a little bit of an element of playing with fire, because you've got this one ingredient and you've got a second ingredient with the ability to turn into something absolutely terrible from the standpoint of the cell's health, but you've got just enough of these natural defenses to make sure that these 2 never meet. And so in a healthy system, we have a relatively even ratio of reactive oxygen species and the trash men to neutralize them, with a very solid barrier between that reactive oxygen species and any iron within the cell, whether that iron be free iron or being actively used within the complexes of the electron transport chain. But what happens when we take on too much free iron and you start to see these little iron molecules accumulating within the cells? Well, all of this free iron within the cell would probably want to go to the mitochondria, wouldn't they? Because that's where they know they're needed in order to make energy. But what happens when that too much iron in the cell turns into too much iron in that electron transport chain? You start to have wandering iron within the cell. And you've got all of these little bad boys ready to donate their electrons and interfere with the natural deactivation of those ROS within the mitochondria. So with all this free iron all over the place, these guys are starting to wear it on the chin. And the number of these molecules just so happen to running across a free hydrogen peroxide start to create this buildup of entropy, this buildup of reactivity by creating more and more hydroxyl radicals. The more hydroxyl radicals that result from this chemical reaction, the more these natural defenses really need to be on their game. But they've already been overrun by all of these original ROS. So these guys start to get beaten up. And an unhealthy system results from this increase in ROS, this decrease in trash men, and the introduction of free iron. And this actually increases exponentially because, like I mentioned before, this isn't just hydrogen peroxide and iron. This is something that starts with 1 and makes 2 more. So it's growing way faster. And that positive feedback loop of more ROS, more free iron, more ROS, more free iron